So, as you can already see, our topic of discussion today is a small, very small topic, and that will be the coagulase test. Alright. Coagulase test, if you have not seen my discussions on Staphylococcus, then I would say that you should probably give that a view, or you should have at least some Staphylococcus knowledge, because coagulase test is related to Staphylococcus. Alright. So, with that being said, let's actually talk in details about this coagulase test, which is not much, so this will be a short discussion, and let's get everything correctly this time sorted out, and let's understand what this test is all about. So, the first thing that we actually talk about is the very basis or the fundamental of this test, which is the coagulase enzyme. What exactly are you doing in the coagulase test? That is the procedure of the coagulase test that we'll talk about later. But what exactly is the motive, or what exactly is the basis of this coagulase test? That, or rather the answer to that, is the coagulase enzyme. So what exactly is this coagulase enzyme? Something that we have kind of talked about in the Staphylococcus discussions, but we need a little bit overview of this. Coagulase enzyme, as its name suggests, it brings about the clotting of human or rabbit plasma as well. Alright, so coagulation, obviously similar sounding, so you can already kind of figure that out. What this coagulase enzyme does is very interesting. It reacts with something known as the coagulase reacting factor. Coagulase reacting factor, CRF, this is actually present in human plasma or rabbit plasma, but it is not present in plasma of certain animals, one example of that being guinea pig. Guinea pig plasma does not have CRF, coagulase reacting factor. Coagulase reacting factor is a protein, obviously you can see. Like the millions of proteins and peptides present in our plasma, CRF is one of them. With this CRF, the coagulase enzyme, which belongs to the bacteria. Coagulase enzyme is not an enzyme in us or in the rabbits. It is an enzyme which is secreted by the bacteria. What bacteria? In this case, staphylococcal bacteria. Okay. So that bacteria, they will actually secrete these enzymes, coagulase enzyme, leading to actually the clotting of the area. Okay. How that happens, we'll talk about that in the next slide. But this is just a general overview of the coagulase enzyme. Coagulase enzyme has 8 serotypes, and the human strains of Staphylococcus, they mostly have type A coagulase enzyme. This is just bland facts, so there is not much to explain in that. However, what there is to explain about is how does it work? How does this coagulase enzyme work? It binds with CRF, which is something that I've already mentioned before, right? Coagulase reacting factor. After binding with coagulase reacting factor, together they lead to a prothrombin-like activity. This complex, this coagulase reacting factor complex, this has a prothrombin-like activity. Now, from our prior knowledge of physiology, hematology, blood clotting, we know that prothrombin is a very important constituent as far as the blood clotting is concerned. So this prothrombin-like activity will enable this coagulase enzyme to convert fibrinogen to fibrin. And we already know that fibrin monomers, fibrin threads, they will lead to clotting. So that is how the clot is formed, and that is how the coagulase enzyme leads to the clotting around the site of infection. What is the purpose of this? Why is the bacteria doing this? This is because this helps the bacteria in localizing the lesion at the site of infection. Alright. So this is how the coagulase enzyme works, and we have already talked a little bit about the coagulase enzyme itself. Now, let's talk about the different types of coagulase itself. See, I was telling that coagulase, this reacts with coagulase reacting factor and all of that, right? But to be a little bit more precise, this is actually the free form of coagulase. Alright? So only the free form of coagulase is actually reacting with coagulase reacting factor, and as we will see in the later slides, this free form of coagulase enzyme is actually detected by a test, which is the tube coagulase test. Our topic of discussion is coagulase test, but coagulase test has two types or two subtypes: tube coagulase and slide coagulase. In tube coagulase test, we do it in a tube. Slide coagulase, we do it on a slide. We'll see it in a bit. Why I'm telling this is to draw that point into this discussion, that the free form of coagulase enzyme is actually detected by tube coagulase test, whereas the bound form of coagulase enzyme is known as clumping factor. So if you have said that coagulase enzyme does this, coagulase enzyme does that, you can kind of automatically reset your thinking to free form of coagulase enzyme does this or free form of coagulase enzyme does that. What I am trying to mean by this is, whenever you are just said coagulase, you can almost be sure that you are talking about the free form of coagulase, because the bound form of coagulase is called clumping factor and not just coagulase. Okay? This clumping factor does not react with CRF, and this bound form or the clumping factor, this is actually detected by the slide coagulase test. So all of this that we were talking about, this was the free form of coagulase enzyme, but the bound form, this actually does not react with CRF, coagulase reacting factor, and this bound form is detected by the slide coagulase test. Now let's get on with the actual coagulase test itself, which is our point of discussion. What are the materials required for this? For both tube coagulase and slide coagulase test, you need two things common. The first thing is you need the bacteria, and the second thing is you need a sterile saline. Okay? Why we need the bacteria? Of course, we need the bacteria so that we can test the enzymes, whether that bacteria has these enzymes or not. And if it does have, then we can conclude that yes, it is Staphylococcus this or that. We'll see to that. Okay? So 18 to 24 hours growth culture. This means growth culture, so that obviously means that you're talking about the bacteria. So you need an 18 to 24 hours growth culture of the bacteria, and what you need is a sterile saline. You know saline salt water? Sterile has to be. Why you need saline? This is because I had talked about this earlier. This is because saline actually ensures that no other bacteria can interfere with the test, but Staphylococcus, it can actually grow even in saline. So that actually isolates the staphylococcal bacteria, and the staphylococcal bacteria, we can kind of confirm it. Otherwise the test might be false positive, false negative, whatever might happen, other bacteria might interrupt, right? That's why you need saline. Okay? Now these are the general requirements. What about tube coagulase test requiring specifically a 1:6 diluted plasma and sterile test tubes? Sterile test tubes, there is nothing to talk about, obviously. What do I mean by 1:6 diluted plasma? This is something that we will kind of discuss when we'll talk about the tube coagulase test in a bit. Okay? Okay. What do you need specifically for slide coagulase test? You need obviously the human plasma itself. In both cases you need human plasma, but in tube coagulase test, see the difference? You need diluted plasma, 1:6 ratio. But in slide coagulase test, you need pooled clear, undiluted human plasma. A basic difference, alright. Obviously, you will need clean slides and you will need applicator sticks. What are applicator sticks meant for? They are meant for taking a small sample from the growth culture of the bacteria and emulsifying that with the plasma. Okay, we will see as to how they are and the entire procedure is happening. Everything will be crystal clear. Now let's jump into the procedure for slide coagulase test. Let's get on with the slide coagulase test first. Okay. So the first step is that we take a clean slide, clean obviously, and on that slide we mark T on one end and C on the other end, as you can see in the picture. Okay. T will stand for test and C will stand for control. So we are actually testing out, like any experiment, we are making a test sample and we will be making a control sample. Okay. Now we'll draw two circles on the T and the C mark, which will actually kind of demarcate their boundaries. Okay, these circles will actually demarcate the boundaries within which the entire samples will be put, the samples will be emulsified and everything will be done. Okay. After drawing the two circles, we will add a drop of saline in each of the T and the C. Okay, both those circles, in both those circles we will drop saline, one drop of saline. Okay. Now using the applicator sticks, we will gently take out a little bit of the sample from the 18 to 24 growth culture bacterial colony and gently emulsify them in both the drops of saline. So both in the T and in the C. As you can see in the picture, the T has already been done, the emulsification has already been done, and you can see that it has become cloudy white because we have mixed the bacteria in the sample. In the C, we are about to do that. That's when the picture has been taken. Okay. Okay. Now you can see that in both T and C, they both have cloudy white because the bacteria have been mixed in both of them. Okay. Now what we do is we add a drop of undiluted human plasma to the test sample. We do not drop, or rather we do not add the plasma to the control, because that is the entire point of a control, that you do not actually replicate everything that you are doing in the test. You are only seeing if the reagents themselves give any reaction or not. That's what a control is meant for. The second point that you should note from this is undiluted human plasma. In slide coagulase test, you actually add a drop of undiluted human plasma, not diluted. Diluted will be in the tube coagulase. In this case, undiluted. Okay. After dropping or after adding that to the test sample, we take a drop of saline and then we add it to the control sample. Again, we are actually trying to prove that it is the human plasma and the bacteria which are reacting, and not the saline and the bacteria which are reacting to give the reaction. That is why we are adding a drop of saline to the control sample. That is the entire point of a control. We know this from the basics of science, right? Finally, all of this is done, and to mix the constituents gently, we will rock the slide on both sides and the constituents will mix well within each other and the reactions can occur. This reaction in slide coagulase test, it's very rapid, and within 10 to 15 seconds, we will be able to see a change if, or rather provided, that the bacteria is slide coagulase positive. What is the change that we will be able to observe in the test? We will see small clumps are seen. As you can see in the T circle, whitish dots have appeared, whereas in the C, nothing, is the same as before. This proves that the bacteria is slide coagulase positive, or scientifically speaking, the bacteria has clumping factor. The bacteria has coagulase enzyme in the bound form. So it is slide coagulase positive. Okay. Conversely, we can also say, when we will talk about the examples of these bacteria which give these tests positive, we will also be able to deduce what kind of bacteria. So now can you understand why this is a very important diagnostic test? We can detect, we can see whether this is a staphylococcal bacteria or not. Even then we will also be able to distinguish in between different staphylococcal bacteria. How so? When we will talk about the examples of each of these, who gives tube coagulase positive, who gives slide coagulase positive, we will talk about it there. Okay. So I hope the procedure is crystal clear. Now let's move on to the procedure for the tube coagulase test. Okay. So the first step is preparing the 1:6 diluted plasma. Now let me explain to you what this means. The first thing is, forget about 1:6, just concentrate on diluted plasma. Concentrate on diluted plasma, pun intended. So anyways, what do I mean by diluted plasma? Diluted plasma means it's not pure plasma, unlike the previous one where it was pure plasma. Diluted plasma. What are you diluting the plasma with? We are diluting the plasma with saline. How so? We are adding 5 ml saline solution to 1 ml of human plasma, and we are making a solution. So obviously 1 part human plasma in total 6 parts of solution means it is a 1:6 diluted plasma. Are you getting me how it is 6 parts? 1 ml of human plasma, 5 ml of saline solution, total 6 ml of entire solution. Within that you only have 1 ml human plasma, so the dilution factor is 1:6. This is how we prepare 1:6 diluted plasma. Now, as you can see in these two pictures, we take a small sample from the bacterial colony. In the left picture that has been given, that petri dish that he is holding, that is a bacterial colony, that's a growth culture, and he is taking a small sample from their bacterial colony and he is emulsifying that bacterial colony in 0.5 milliliters saline solution or 0.5 milliliters, not millimeter, 0.5 milliliters plasma. Are you getting me? In the left picture, he took the bacteria. In the right picture, he is adding that small amount of bacteria to how much saline solution or how much plasma? 0.5 milliliters plasma. This is not the same test tube as the previous one, this one. Here there isn't any test tube, but what I am trying to say is that here we prepared 1:6 diluted plasma, separate thing. Now we are preparing within this test tube, we are actually emulsifying the bacteria. We are emulsifying the bacteria in what? In 0.5 milliliters of saline, or you can also do 0.5 milliliters of plasma. Okay. Now we add 0.5 milliliters of the 1:6 plasma in this test tube. So what we did, we first made this 1:6 diluted plasma. We prepared it in this container that you can see and we kept it. We marked it 1:6 diluted plasma with a marker and we kept it aside. Then we took a test tube, we took a 18 to 24 hours growth culture bacteria, took a small part of the bacterial colony and emulsified that small part of bacterial colony in 0.5 milliliters of saline or plasma solution inside a test tube. Within this test tube, now we added 0.5 milliliters of this thing, the diluted plasma, 1:6. Okay. Now, this tube, this test tube where 1:6 diluted plasma and the bacterial colony emulsified in the 0.5 ml saline solution or plasma solution. This test tube holding both those solutions, this is actually incubated for 4 hours at 37 degrees Celsius. Finally, the tube is taken out and tilted very gently, and we observe any clot inside. Why do we tilt it? So that the unclotted liquid can flow, the clotted one will be easily differentiated out. Why do we do it gently? So that the clot, which is not that stable, not that strong, does not break off. Okay? If this clot is actually seen, voila, it is tube coagulase positive. What does tube coagulase test actually test? It tests for the free form of the coagulase enzyme. So, if the bacterial colony that you actually emulsified, inoculated, if that actually produces this coagulase enzyme in its free form, very good, you give tube coagulase positive. So, the question is, who are these bacteria which give these tests positive? All of them, obviously they are Staphylococci. Staphylococci, this is a test unique for Staphylococcus, obviously. That is why we study this under Staphylococcus. So obviously this test, tube coagulase, slide coagulase, this is the most important diagnostic test for differentiating Staphylococci from any other infection or from any other suspected bacteria. But even within these Staphylococci, we can see that both slide coagulase and tube coagulase positive is given by Staphylococcus aureus, the most important and more pathogenic Staphylococcus, Staphylococcus haicus and Staphylococcus intermedius. Only slide coagulase positive, not tube coagulase, only slide coagulase positive. This is a very important example, Staphylococcus lugdunensis. And only tube coagulase positive, Staphylococcus delphini. So these are more or less, these are bland examples, but I would really draw your attention towards the fact that Staphylococcus aureus, if you don't remember anything, only remember the fact that Staphylococcus aureus is both slide and tube coagulase positive. Finally, we arrive at certain differences. What are these differences in between the tube coagulase and slide coagulase test? Tube coagulase, as I have already mentioned, is due to the coagulase enzyme itself. Slide coagulase, on the other hand, is due to the clumping factor, which is nothing but the bound form of coagulase enzyme. The free form in tube coagulase requires CRF, coagulase reacting factor, as I have mentioned, but slide coagulase does not require CRF. It automatically actually has a prothrombin-like activity. It will automatically lead to fibrin threads from fibrinogen. Tube is done in a tube, slide is done on a slide, as the name suggests. That's a very basic and obvious difference. Tube coagulase, in that a clot is formed, and in slide clumps are formed. Now these are differences which are, I would say that if you have a lot of like asking marks, only then go for these differences. These are very like bland and basic differences. Okay, so I wouldn't really recommend differences from 4th point onwards. Anyways, the coagulase enzyme itself has 8 serotypes, whereas clumping factor has only 1 serotype. See, what kind of a difference is that? That's a very weird difference. That's like saying that an elephant has a tail, but a goose has wings. That kind of a difference. No, that's not a difference. What are you talking about? Anyways, so however, the next difference is very important, and that is why I said that this one is very important that you remember, that Staphylococcus lugdunensis gives a negative tube coagulase but gives a positive slide coagulase. It's only slide coagulase positive, whereas both tube and slide coagulase positive for Staphylococcus aureus, haicus and intermedius. Okay. So with that, we end coagulase. This was a small discussion, not much to have said, but this was an important discussion, I think, because this actually is very, very relevant to the lab diagnosis and the most important diagnostic test for Staphylococcus aureus. So with this, we come to an end, and I do hope that you found this useful. Thank you, and we'll meet again in the next discussion.